This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 478, recorded on January 26th, 2018. Hi, everybody. I'm Vincent Racaniello, and you are listening to the podcast all about viruses, coming to you from New York City, where it is sunny and 2 degrees Celsius. Joining me today from southeastern Michigan, Kathy Spindler.
Hi, everybody. I didn't know that Dickson wasn't there, and so now I
threw off your game, right?
The weather is now up. It's 49 degrees. They said it would be close to 50 today, and so it is. That's Fahrenheit, which means it's 8 degrees Celsius, and it's sunny, wispy clouds. It's lovely.
This morning, the Hudson River was not a ripple on it. Usually there's some kind of chop and ripple.
Wow. Like
glass, and the bridge was reflected in it.
Oh, neat.
I took a picture. I will send all of you. Dickson is teaching his course. He'll be in later.
Oh, right.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And the National Weather Service describes the weather as fair, but I would say it's good.
Fair.
It's 35 to Celsius. Lovely, sunny, just a few puffy clouds, light southeast wind. Very, very nice for this time of year.
And from Austin, Texas, Rich Condit.
Hi, everybody. We got 61 degrees. That's 16C. And high overcast or cloudy or something. So it's kind of gray,
Kind of gray.
But that's okay. I'm in here doing TWiV.
And Dickson will be in later. Hey, Kathy, what's up with ASV?
Oh, I have to switch back now. ASV, you only have 4 days to get your abstract in. And we're starting to have people who are messing that up. So make sure you like how you have your abstract before you push the final submit button. I think you can go back and edit it more than one time. Anyway, this won't be relevant for most people. But I did want to say that we haven't yet publicized the fact that we're having what I've just decided to call a boot camp for assistant professors. There'll be a dinner on Sunday night to provide assistance and support to recently hired assistant professors. There'll be hosted tables with topics about federal funding, hosted by program officers from federal agencies, managing people and money, navigating tenure, and advice from the recently tenured. So this is a new thing that we've added, as if there aren't enough career and other things going on at ASV. But we think this will be good for new assistant professors. So we haven't yet publicized it, but I'm pretty sure it'll be just pretty much open to people that want to do it. And as I said, it's on the Sunday night of the ASV meeting.
All right. 4 days. Get those abstracts in. I'm one of those that we have to get ours in. Mm-hmm. We'll get it in next week. Mm-hmm. All right. A couple of follow-ups. We're going to give away a book later because nobody sent in a crossword. So I'm going to try giving it away. Just send in your email. Okay. Follow-up from Islam. I love TWiV. Thanks to you, Vincent, and all the members of the TWiV team. This is the best educational experience ever. I have reached a stage of TWiV addiction where a paper is not a paper until I hear it discussed on TWiV, and I would love to hear every single new virology-related finding discussed by you guys because I learn a lot. For those who want a shorter version of TWiV, I'm listening to the ARC episode for the second time in the same day. Wow. Islam Hussein from Viral Vlog. Okay, next. Last time we had an email from Daniel who sent a link to a TEDx video wherein Dr. Andrei Goodkoff said 2 things which Daniel asked us about. First, did retroviruses cause the post-dinosaur diversification of mammals, and are endogenous retroviruses in our genome responsible for aging? So I went and looked at it. He got it all wrong. Now, first, he spends a lot of time talking about aging, and that where he doesn't even mention a virus. And then he said aging is due to senescence of cells. And he says, why is that? He said they must have a clock, but he never mentions telomeres, okay, which are important in senescence and dying cells. And then he says half of our genome is viral, which isn't true. It's not true at all. About 8% are LTR retrotransposons, but not all of those are even endogenous retroviruses. So it's less than 8%. The other stuff is repeated sequences, lines and signs, and various elements, mobile elements. Okay, so it's not 50%. Then he says, after the dinosaurs left, that's when retroviruses came on the scene, and they infected all the mammals, and he thinks that was responsible for their diversification, of course, for which there's no evidence, and he provides none.
And there's actually counter evidence because there's retroviral integration in non-mammalian lineages.
And that's the other thing. We can now date it to 450 million years ago, just at the end of the Cambrian explosion. We did that paper here on TWiV. Mm-hmm. Phylogenomics indicates that they were around a long time ago, probably originated in the oceans, if not before. So they didn't just start. And so there's no evidence for his hypothesis. And he says here's the one thing he does show. He says our retroviruses are pretty silent. And his idea is that they get more active as we age, but that's not true. By silent, I suppose he means they're not making infectious particles. And he says, in mice, they do make infectious particles, and that's why mice only live 2 to 3 years. And we live longer because ours are silent until we get old. So this is totally bogus. Totally bogus. And
TED Talks really went downhill.
I was just going to say that. I just don't like them, especially TEDx's, right? They're not even vetted.
Right.
So, they're just
YouTube videos, basically.
They're YouTube videos, really. This is TEDx Morristown. I can see getting, Carl Sagan to do a TED video. That would be fine. But
that'd be amazing.
Yeah, that'd be amazing these days. But,
I'd be very impressed.
Some Phil Sharp or something like that. But I don't know who this guy is, and he makes these claims, and people are laughing, and they're interested, and so I think they're doing a disservice. It's just another YouTube video. You just don't know. In the old days, you could say, "Oh, it's a TED movie, a TED video. It's probably good," but you can't even say that anymore. Right. So that's my thoughts about it.
Well, that's too bad.
You would like retroviruses to be responsible for your senescence?
No, as a matter of fact, I don't understand all of the fuss about aging. Okay, this happens. Fine.
You know what he says? He said we get to about 120. That's the most we can go to. And it's like, how do we get beyond it? Why do you want to get beyond it?
Yeah, exactly. Who cares?
I think 90 is pretty good, or 80 even, right?
Jeez, I'm happy to be the age I am. It's good.
Yeah. Yeah, I know. There's a lot.
So I don't understand that, and it's disappointing to have just bad stuff, wrong stuff out there.
And if we know anything from fantasy fiction, it's that immortality is a curse, right? You'll end up in a situation you can't escape, and you won't die, and you'll be stuck there forever.
Yeah, the vampires don't like
bricked in
forever.
A wall or something.
Yeah, got to get blood meals every night.
Yeah.
Now, if you read the Anne Rice books, that's what the vampires complain about. They can't. They're always around. They can't die. Yeah. Anyway, so that's that, Daniel, if you're listening. And now, Alan, you found a little snippet at the last minute. You want to tell us about that?
Yes. So this is from MMWR, which is back online.
It was offline?
And it wasn't offline, but I don't know if it would have been published on schedule if the shutdown had continued. In fact, I suspect it wouldn't have.
Probably not.
Anyway, this is from Morbidity and Mortality Weekly Report, MMWR, which if you don't already have it in your news feed, you really should, because it's got just really neat stuff coming out all the time. And it's not too high volume either. But this is a population-based surveillance of birth defects that could be related to Zika virus infection. Now, being the CDC, they have these registries, birth defects registry, and the ability to collate data from it. And this thing has a gazillion authors on it. First author is Augustina Delaney, and senior author is Margaret Honan. And they're from a bunch of different places, and they're MDs and MPHs and PhDs, et cetera. But the basic concept here is that they monitored for birth defects that could be associated with Zika virus infection. So they're looking for things like microcephaly and the other things that have been associated with Zika in northeastern Brazil. And the enduring mystery with this is that in subsequent Zika outbreaks, in many cases, we don't see this spike in birth defects. So what happened in northeastern Brazil? Was it weird, or was it just that we're not looking closely enough? This paper, which is open access, you can go check it out for yourself, and it's quite readable. They look at, they've got 15 states and territories. So they looked at Puerto Rico, but they also looked at a bunch of southeastern U.S. states where there have been imported or locally transmitted Zika cases. And they did, they looked at before and after. So looking, let's see, I guess it's first half of 2016. And then they compared that to after introduction of Zika virus into the U.S. and looked at the rate of birth defects that could be Zika associated. So they've got huge numbers. N is enormous in this. It's I misplaced it now in the document, but it's basically all the whole birth cohort in this 15 state region. And they're looking at all of the birth defects in that cohort and the subset that could be Zika associated. And they found that, in fact, potentially Zika associated birth defects did go up after the introduction of Zika. So if you have enough, if N is big enough, you can actually detect a possible signal from this. And that's really, really interesting, I think.
That's why Brazil was so obvious, because it was huge.
Yeah, right. And it's why it wasn't obvious before, because the populations were small.
Right.
In the sidebar, they mention a birth cohort of nearly 1 million births.
There's where it is, yes.
1 million.
1 million births.
I think somewhere I remember that 7,000 of those had evidence of Zika infection.
Yeah, the actual birth defects numbers, the numbers end up being small, like in the
3 per thousand.
3 per thousand.
Due to Zika.
Right. But, those are excess cases that didn't occur before the virus arrived. And so it's not conclusive, but it is very, very suggestive of link.
29 more cases than expected.
29 more cases than expected.
Yeah. No, it just has to be done to figure out because that's the question everyone asks, what's happening in the U.S. where we did have some, right, imported and endogenous or autochthonous. So this is good. Yeah, very good.
Yeah.
All right. Going along with that, we had an email from Neva from Buda, Texas. This is not far from Austin.
I had lunch with Neva last week, and I saw her at the march here in Austin, the Women's March last week.
Cool. This is in the Texas Observer, article called Bordering on an Outbreak by Sophie Novak, Brownsville, Texas, way down there on the border, right?
Yep.
Can't afford a Zika outbreak, but it also can't afford to prevent one. It's kind of a sad story of how, that's the place where there was an outbreak, right, in Brownsville.
Mm-hmm.
That was the other locally transmitted Zika, in addition to Florida, Miami. And they've had dengue here before, and they don't have money for health care. The people don't know much about diseases. This one woman cited in the paper, in the article, says, I thought I was supposed to put mosquito repellent on my belly only, and she was pregnant and not elsewhere. So they don't have education to teach people this. So healthcare system is pretty strained. And, this is
very strained.
You can do all you want with biology, but if you don't have a good healthcare system, it's a big problem.
And this is a very well reported and well written article that is well worth your time. It came out in December, but it's I hadn't seen it, and it's the story of public health. It's just the political impetus is, who cares what happens to poor people, and we're not going to deal with anything that's not a crisis. And so this is where your crises come from, because these are the people who are going to get sick, and it's going to spread to everybody. This will be everybody's problem, whether no matter what. and yet, the funding for Zika is drying up because it's no longer an acute crisis. It's not in the headlines anymore. And these are the people, the public health workers who Novak is talking to in the article are the people who are at the front lines of this, dealing with it every day. It's a really, if you haven't tracked this issue or you've lost touch with it, it's really worth going back into this.
Public perception is that Washington and Austin have earmarked millions of dollars for Zika, but we're not seeing any of it.
Yep.
So in my opinion, this is one of the functions of government, to provide public health and health care for the populace. And not everyone believes that, unfortunately. And it's just, in my opinion, that's not right.
Yeah.
It's our job. It's government job to do that. I firmly believe it. And, it's getting harder and harder to do.
Here in Texas, we'd rather spend our legislative time talking about who can use what bathroom.
Yeah.
Yeah. Yep.
And no income tax. So there's your funding.
All right, we have a main topic today. And
yes, we do.
I just want to say that it has to do with viruses. Are you surprised? Well, this is an article, and Rich will tell us all about it, but I had gotten an email from Richard, DDS, a dentist who follows us, and he sent an article published in NTI. It's called, and the website subtitle is Building a Safer World, and it's an article by Dr. Elizabeth Cameron, which is entitled Horsepox Research Accentuates Urgency for Global Action to Reduce Biological Risks. And this is all about a paper that has come out in PLOS One. And I think we'll turn to this at the end of our discussion. But I'm going to hand it over to Rich Condit, because if it's pox virus, it's Rich Condit.
And by the way, the NTI is the Nuclear Threat Initiative.
But what's this atomic pulse underneath? I guess that's just like a subset of their global energy, nuclear policy, right. Nuclear terrorism, cyber, biosecurity.
So NTI was founded, I'm just looking at the About Us page, in 2001 by former U.S. Senators Sam Nunn and Ted Turner. Mm-hmm. And it's I guess it's a nonprofit that advocates for
restricting research.
Well, their main thing is preventing the spread of nuclear and biological weapons and chemical weapons, and so they're like a weapons anti-proliferation group.
Mm-hmm. All right, we'll come back to it. Meanwhile, this is the focus of this TWiV is a paper published in PLOS One, and I'm going to hand it over to Rich to tell us all about it.
Okay. So I want to start off, actually, as I also recommended a snippet about a smallpox vaccine based on horsepox. And what I want to do is give the background leading up through the snippet to the paper. So I want to emphasize why this is important. First of all, it's history, and it's fascinating history. It relates to the most successful vaccination story that we have, which is the eradication of smallpox. Which was done before people knew what a virus was or anything about immunology or anything. So it was done in a vacuum of knowledge, and we're only beginning to understand, looking back, what in fact was actually done. And it actually constitutes a pretty interesting mystery.
I would say it started before we knew all this. The eradication effort, we did know a fair amount about virology at that point.
True. Yes. The information that we get out of this might be useful in my opinion, low probability event of smallpox returning, but nevertheless, it would be useful under those circumstances. I think what we learned from this may be useful for understanding vaccination in other circumstances in the future. And as we'll see, the technology itself, ultimately in this paper in PLOS One, is fascinating. So a very quick review of smallpox. It is, when I think of the word pestilence, I see smallpox. It is probably the nastiest of the nastiest diseases that you can imagine. You're covered with painful, blistering sores. You got a 25% chance of dying of it. You're going to be scarred and have other problems if you manage to survive. It was prevalent in all the major population centers in the world in the 14th, 15th, 16th, 17th, 18th centuries, to be sure, until it started to disappear because of vaccination, and probably before. So if you lived in Paris or London in the 17th, 16th, 17th century, you knew your kids were going to get this. It was only a question of whether or not they were going to survive. And it's really nasty. We probably ought to have a picture of smallpox for the episode picture just to remind people of how nasty it is. So, a brief review of the history of vaccination. In 1796, Edward Jenner, capitalizing on some observations of others unpublished, documented officially that if you had been infected with cowpox, which was a disease mostly of people who worked with farm animals, in particular milkers of cows, they'd get these blistery lesions on their hands, that you were resistant to smallpox. And he ultimately did the experiment of taking, it turns out, material from the hand of a milker that was one of these lesions, and scratching it into the skin of a young boy, and then later using the technique of variolation, where you would actually immunize people using the smallpox virus itself to challenge the kid with smallpox. And he was resistant to that procedure. So he proved that this material that came from cows could be used to confer resistance to smallpox. And this practice spread worldwide until ultimately in 1978, 1980, smallpox was declared eradicated. Now,
and we have a TWiV on this, right? Where we went through Jenner's original report, and
as a matter of fact, we did. We did Jenner's actual inquiry, and I just to emphasize this emphasis on cowpox, I want to read the full title of Jenner's inquiry. It's called "An Inquiry into the Causes and Effects of the Varioli Vaccine, a Disease Discovered in Some of the Western Counties of England, particularly Gloucester, and known by the name of the cowpox." So basically, everybody assumed for the longest time that this was cowpox.
There's just one problem.
Yeah.
It's not.
In 1939, a guy named Downey, now this is just at when virology was ramping up globally, compared vaccinia. Okay, so the virus that is in the smallpox vaccine is called vaccinia. Compared vaccinia to what was then known as cowpox, using a variety of different comparative. He obviously didn't have any genetics, but he could do serology and some other characterizations, and came to the conclusion that the smallpox vaccine, vaccinia, is not cowpox. Not only that, but there are no known naturally occurring viruses that resemble vaccinia. So this precipitates a mystery. What is the vaccine and where did it come from? And this has been a long-standing mystery.
The greatest mystery in virology, really. With this incredibly successful vaccine that's been used for centuries. It is the first vaccine. It's a landmark in medicine and in microbiology, and it was used for the first eradication of a virus. I mean, this is incredibly significant, and we have no idea where it came from.
Right.
But it all started with Jenner, though. Is that
it started with?
It all started with Jenner.
We don't know what virus. Where did this?
It works.
Where did you get the virus?
It works.
It works.
Does, and that's great.
Yeah, but so that's one of the relevances of this. It works, but we don't know where it came from. Mm-hmm. So why does it work?
Do you think that would be licensed today?
Oh no, his procedure, no.
Well, and in fact, the original vaccine was loaded with bacteria.
Yeah.
Mm-hmm.
Right. And in fact, that was we discussed Tort's work and Durrell's work finding plaques, and one of the original source materials that they used was the smallpox vaccine, because you could just plate it out and you got a lawn of bacteria and it grows phage out of it.
So at any rate, this has remained a mystery for a long time.
Put another way, Rich, the virus you spent your life on, you have no idea where it came from.
Right.
Yeah, but we're finding out. So one of the seminal treatises on this mystery is a 1981 publication by Derek Baxby, who proposed, now there were a lot of proposals, but this one becomes relevant, proposed that in fact vaccinia was derived from horsepox, a virus that even at that time, it was essentially extinct at that point. His conclusion that it was horsepox is based primarily on historical records. And in fact, if you go all the way back to Jenner, what you find out is that Jenner thought that people didn't, because there was all this emphasis on cowpox, people sort of brushed this under the carpet. But Jenner thought that in fact the active ingredient that he was working with came from horses, from a blistering disease in horses called grease.
Right.
But that Jenner thought that it actually had to be passaged through cows to become fully potent and useful against smallpox. And the way this would happen was people who handled the horses also handled the cattle. And so there would be human transmission of this agent from the horses to the cattle, and then from the cattle to other individuals, and that would confer resistance to smallpox from them. Now, as it
Rich,
turns,
in this paper of Clarissa's, this review, they have nice pictures of people with blisters on their hands and face who worked with horses and cows. Mm-hmm. It's really, I'd never seen those besides these Jenner drawings before. They're really good.
And specifically, they talk about the horse care people being farriers who specialize in hoof care.
Yeah.
Mm-hmm. Right.
Which is where the grease shows up. Yeah.
Shows up. Yeah.
So if you then look at the history of vaccine production, and this, we can link two different reviews, one by Clarissa, and let me get the other one up here because the authorship is important. Another by Jose Esparza, Livia Schrick, Clarissa Damaso again, and Andreas Nietzsche. And these are primarily historical reviews looking at where vaccine came from and how it was produced. And the bottom line is that there is ample evidence that in many cases, people were using material that came from horses. They were using horsepox. There's demonstration that horsepox itself works as a vaccine, and there's lots of evidence that people derive vaccines from horses. Now, it's a lot more complicated than that because there's also a lot of evidence that people used material from cows, and at various times people would mix vaccine sources from various sources. So, and none of this, of course, was regulated or controlled until much later in the smallpox eradication campaign, when the World Health Organization identified a few different sources of vaccine and said, I think there were 4, and said these are going to be it. And most of the modern vaccines are derived from those. Now, one of the important ones that Clarissa discusses in her review, I'm going to butcher the name here. Would you pronounce this Beaugency?
Beaugency.
Beaugency.
Mm-hmm.
She describes the Beaugency lymph or vaccine, which was fairly well characterized and obviously has a component that, well, at least from the history, could have had a component from horses. And she was interested in this because of her own research into the Brazilian vaccine, and the vaccine escaped into the wild in the form of Cantagallo virus. And she and collaborators characterized the Brazilian smallpox vaccine, the IOC vaccine, Institute Oswald Cruz vaccine, which they knew from good historical records came from the Beaugency lymph. And that vaccine, the virus in that vaccine, its closest known relative is horsepox. Okay, so or at least horsepox is the closest ancestor to that. So there's a direct line from a well-circulated, well-characterized vaccine to horsepox. Mm-hmm. Now we come to the snippet. Go ahead.
Well, I was just going to say, before we get too far, because this is called Beaugency lymph, and lymph might connote something to a number of people as being a component of blood. In this case, the lymph is really lesions that were ground in a mortar and suspended in diluent, and that was considered lymph. So it had nothing to do with something coming from animal or human blood. It was just from the lesions, the pox, as it were, being ground up and diluted.
And also, Jenner had done this. He had taken horsepox lesions, right, and inoculated people sometimes, and he called it equination, or someone called it equination. I like that.
Right. And there are numerous other examples of the fact that you can take horsepox lesions from horses and get the same effect as you would with smallpox.
This review by Clarissa, I don't know if it's open access, but it's really good because it gives you this idea of how everyone is swapping their stuff with everyone else all over the world. I mean, every country wanted this, and no wonder it's a mess, right?
Right.
So that brings us to the snippet, okay, which is, to me, just fascinating, which is an article in the New England Journal of Medicine. First author is Livia Schrick, and other authors are Simon Tausch, P. Volchek Dabrowski, Clarissa Damaso, Jose Esparza, and Andreas Nietzsche. And there's a burgeoning collaboration here with Clarissa in Brazil, Jose at the University of Maryland School of Medicine, and Andreas Nietzsche and his collaborators, Livia Schrick and others, at the Robert Koch Institute in Germany. So this is kind of a hobby for these. This is not their day job, really. Okay, but they're all fascinated by this and all contribute their own bits. Jose is, I would say, obsessed with the history of the vaccine. He's really a fascinating guy. I've had a lot of correspondence with him. Andreas has an interest in pox viruses, but also a very robust sequencing effort. So he's done a lot of the sequencing. And Clarissa has a long and deep history in pox viruses and an interest in the history and her own interest in the Brazilian vaccine strains and escaped vaccine strains in particular. So it's a delightful collaboration. So Esparza's interest in tracing the origins of the vaccine are focused recently on collecting as many different vaccines as he can, okay, and getting sequence on them to see what's in them. And this one describes the sequence of a vaccine sample that was produced by a company called Mulford in 1902. And they managed to get some DNA out of it and sequence it. And lo and behold, it is essentially horsepox. It's got a couple of deletions in the end that show up in vaccinia. So it has evidence of drift towards the more modern vaccines that we know, but its closest virus in the database is horsepox. And a little interesting aside, he got this vaccine on eBay.
It's amazing.
I was gonna say, where did they get this?
Amazing that he got the whole genome sequence out of this. It's amazing that this thing, and he's a picture of the case, came with a scarifier, I guess, right?
Yeah, it looks like. I don't know exactly how this is constituted.
This little wooden cigar case.
Yeah, and there was a vial in it, and I think a capillary with this. It was in a glass capillary, which is good, and a scarifier because it says case and scarifier. Keep it a cool place. eBay.
He's gotten several other samples off of eBay that he is pursuing. And in fact, in my correspondence with him, he's hoping that maybe his vaccine collection effort will get a TWiV bump. Okay. Yeah. So any of you listeners out there who know anything or have any access to vaccines, museum curators in particular, those types, if you got anything that looks promising, contact Jose. He'll be interested. If they can, they'll sequence it. At any rate,
I'm looking at eBay. I don't see any vaccines here.
I looked on. I couldn't see any.
My gosh.
So at any rate, there is really direct evidence that at least one of the American vaccines used was in fact horsepox.
It occurs to me that this stuff's probably still good.
Yeah. It must be.
Right? I mean, they got full DNA sequence.
It could be. It could be. Yeah, it could be.
I'm not advocating anybody do that, but
right. I think that they have, there's lots of samples that they've gone after that they can't get anything out of. Okay. So they were lucky with this one, though it is quite a stable virus.
By the way, I was in this article of Clarissa's. She talks about this Mulford company, and then she has this little part here. Farms in Clifton and Patterson, New Jersey were used for propagation of the animal lymph. And this is in the late 1800s. And I just want to say, and then after 1884, the vaccine was mostly produced by a vaccine farm located in the back of the health department on Mott Street in New York City. Because right now, Mott Street is the village. You're not going to get any farms there. But the cool thing is
that used to be down near the meatpacking district.
Yeah.
It looks like it's in Chinatown.
Chinatown.
Oh, Chinatown. Okay.
I was born in Patterson, New Jersey. And Clifton, my dad had his doctor's office in Clifton. So I know these places really well. And there's no farms there anymore, of course, but it used to be farms, just like there were farms in New York. So I thought that was cool. Just wanted to mention that. Plug for Patterson.
a couple of words about horsepox. It used to be reasonably prevalent in Europe and Asia, but it is apparently, as far as we know now, extinct. We have one sample that came from an outbreak in Mongolia in 1976 that was sequenced in about, I forget, I had the paper up here a while ago, about 2000 or something like that. And so that's the only example that we have. So going back to this mystery, we had this virus that was vaccinia, and we didn't know where it came from. Now, based on this work, we can say that the closest relative of a virus that we know was in nature was horsepox. It's still a mystery to me. The evolution of the vaccine, was horsepox maybe much more prevalent? I mean, why that it's clear that in many cases they were using infections in cattle to make vaccine. And yet none of these vaccines that the vaccine experts can correct me if I'm wrong, but there's not a lot of evidence that there's a heavy cowpox component in any of these. The closest relative is horsepox. So maybe horsepox was a lot more. It certainly was a lot more prevalent in Jenner's time and subsequently. Maybe there was a lot more transmission of horsepox between horses and cattle. So maybe a lot of times when people picked stuff up off of cattle, they were actually picking up horsepox. And maybe
that horsepox grew better on cows than cowpox.
Exactly. Maybe as people passaged the vaccine, okay, which ultimately was done in cattle, that what grew out of that was the stuff that grew best, which was more horsepox-like. And then ultimately, as the World Health Organization said, okay, enough of this craziness, we're going to identify these 4 vaccines as the ones that are going to be used from here on out. They identified ones that contain primarily horsepox-derived material. Now, vaccinia is different than horsepox, but the closest relative is horsepox. There's been a lot of evolution of the virus in the hands of people.
Back on the idea of horsepox maybe growing better in cows or vice versa, the primary reservoirs for these viruses are, at least with respect to cowpox, seem to be rodents, and it's speculated that maybe rodents also for horsepox. So the fact that they're in horses or cows may have to do with the fact that, for instance, cowpox has a really broad host range and things like that. And it reminds me of what we've talked about, for instance, with plant viruses, that a virus gets its name, based on where it was first identified in a plant as a virus, but that doesn't necessarily mean it's the main reservoir or the only place where the virus is.
It's also interesting to consider why horsepox went extinct. Okay.
Yes.
And it could be partly because of the vaccine campaign, partly because of because horsepox was transmitted to some extent. It transmitted to some extent, at least by people, in particular transmission to cows. So as people become vaccinated and as animal husbandry techniques become better, that probably contributes to the extinction.
Also, the number of horses and the crowding of those horses plummeted with the development of the train and the car and the bicycle. So we have a lot fewer horses than we used to. We have more cows than we used to. But then the horses that we do still have tend to be, high-end pets. So they're cared for better.
Clarissa's article also has a very nice phylogenetic tree of the vaccine so that you can see very clearly the relationship. She actually divides the vaccine viruses into 3 different clusters. She calls the American or Dryvax cluster, the South American cluster, and the Eurasian cluster. And there are recognizable vaccines in each of those clusters. The American or Dryvax cluster, of course, contains the viruses that were used in vaccination in the U.S. South American cluster contains the IOC, which was derived from the Beaugency lymph, and the Eurasian cluster contains several that were used in Europe. They're all quite closely related, and horsepox is right in the thick of it.
And then outgrouped from that is cowpox virus, and then outgrouped even further from that is the variola virus or smallpox virus.
Right.
So, Rich, where would a monkeypox fall on this?
Actually, I provided another
there it is
in the show notes.
Yep, I see.
Let me see. Let me go back to that. It's out there. I can't even find. Holy cow!
It's right above. It's right above the one you were just talking about. Yeah, I see.
Yeah, but you know what? I lost.
I see they're all there. Yeah, including Yatapox virus, one of the favorite names.
Yeah,
which Grant McFadden talked about when he was on a long time ago.
Yeah, so monkeypox is there's a group that are called orthopox viruses, and that includes cowpox, variola, monkeypox, vaccinia.
Got it.
And they're all reasonably closely related, but monkeypox sits between variola and vaccinia.
Yeah.
Another probably rodent virus. Okay.
Right. Okay.
So that brings us to the paper.
Yes,
and just in time,
the main paper. These are other papers.
Just in time, Dr. Despommier has entered, so let's let him get seated. We're just starting the main paper.
This is a paper in PLOS One.
You started a horse around?
Yes, authored by Ryan Noyce, Seth Lederman, and David Evans from the Department of Medical Microbiology and Immunology and the Li Ka-shing Institute of Virology at the University of Alberta in Edmonton. Alberta,
where you and I visited, Rich, and did a TWiV a number of years ago, right?
And also from a company called Tonix Pharmaceuticals.
Which is apparently here in New York City.
Yep.
I wonder if it's on the former site of the cowpox manufacturer.
Down there on Mott Street.
Mott Street.
It's probably at Belmont Park.
And because this is in PLOS One, it means it's open access.
Yes.
There you go.
Now, the stated motivation for this is that Tonix Pharmaceuticals, in particular, was interested in exploring the possibility of using horsepox as a smallpox vaccine alternative. Let's take a moment to talk about that. First of all, as we've already described, now in retrospect, horsepox would be the ideal place to look for a smallpox vaccine.
Right.
Because that's probably what it was. It's a good bet that that's what it was to start with anyway. So smallpox has been eradicated. There is always the danger that it could reemerge, either from somebody deliberately. There are two known reservoirs of smallpox, one in Moscow and one in the CDC in Atlanta, both under high security containment. So there's those are the two known sources of smallpox. So there's always the notion that a bioterrorist event or some other event could result in a reemergence of smallpox. The U.S. government has mandated that to protect us against that, we need a good vaccine and we need two antivirals. We have vaccines. The second generation the first generation, the original Dryvax vaccine used in the U.S. would no longer be acceptable by any measure.
It is, however, still given, and it is stockpiled.
They've got the old stuff. What is mostly stockpiled now is a derivative of that. That's a plaque-purified virus called ACAM 2000. Oh. There was a virus that was actually plaque-purified out of the original Dryvax. It has many of these vaccines have side effects. There's about a 1 in a million chance that you'll die if you're the first-time vaccinee from that due to basically an uncontrolled vaccinia infection. And there are other side effects as well. What showed up in a recent effort to vaccinate healthcare professionals was a myocarditis that wound up aborting that campaign. So there are problems with that vaccine. The third generation vaccine is called MVA, or Modified Vaccinia Ankara, which is a vaccine that was developed in Germany, which was a vaccinia that was passaged in chick embryo fibroblasts for 500 passages and accumulated all sorts of damage, deletions, and et cetera, and is so wimpy that it can't go through a complete replication cycle in human cells. You can grow it only on chick cells and BHKs, and in human cells it gets through all of the gene expression but can't assemble particles. That has been grown up and stockpiled. It actually can't even be administered by scratching it into the skin. It has to be injected. But it is safe, and all indications are, based on its use in Germany a long time ago and all its profile in animal models now, that should be effective. That doesn't exclude the possibility that maybe there might be a better vaccine out there. There are some who will argue we don't need another vaccine. Arguments can be made that maybe we should look at alternatives anyway, at any rate, Tonix Pharmaceuticals' motivation is to look at possible vaccine alternatives.
Hey, Rich.
Yeah.
When you immunize people with these various vaccines, what antibodies are the protective antibodies are directed against what viral proteins?
A number of different surface proteins for the most part.
Okay.
Actually, that's another big unknown, and part of this is me not knowing. But the correlates of protection are not fully understood. Okay.
May not just be antibodies, because this is an infectious vaccine. So,
yes, I think I think cellular immunity is part of the part of the program here.
Okay.
Okay. So, Tonix Pharmaceuticals would like horsepox. There's only one source of horsepox, and that's at the CDC, and they are unwilling. They ain't giving it out. In particular, they aren't giving it out for commercial purposes.
By the way, I just want to say, Tonix Pharmaceuticals is a Columbia offshoot. Just so you know, Seth Letterman used to be here. Do you know that name, Dickson?
I do.
So he and Don Landry, who is, I think he's head of medicine or infectious diseases, don't remember. They both started Tonix Pharmaceuticals, medicine.
This morning.
You did?
I did.
And they are, Tonix, the two of them started it. And if you go to the Wikipedia page, they say they're working on various things. And in 2016, they started a biodefense project, and I would guess that this is the biodefense.
You would. By the way, are horses not getting horsepox anymore?
It's apparently extinct.
Extinct. That's weird.
Yep.
Couldn't we have? Oh well.
We talked about it earlier. Sorry, you'll have to listen to TWiV.
I will.
You might like it.
I'd love it.
You might like it.
I know this book that you have in front of you.
It's a gender book. It's lovely.
I have that book. Yeah.
It's a thing to read because you have to transfer all the F's into S's.
Exactly right.
All right, sorry, Rich.
Okay, so I don't know the details of how this came about, but Tonix eventually fell in with David Evans, and Dave says, "No problem, we can make horsepox."
No problem.
Just like Vincent made poliovirus.
No problem, but well, exactly. This is in the realm of synthetic biology. So basically, what the paper does is it synthesizes horsepox in chemically synthesized fragments. There are like 10 overlapping fragments. This is incredible. Some of them up to like 10 kilobases.
They just ordered them too.
Yes. It's incredible that synthetic DNA synthesis has gotten that good, that you can get molecules up to 10 kb or so, where the sequence is okay.
That's for some viruses. That's the whole genome.
Right.
Did you ask him by any chance how much it cost?
No.
I'm just curious how much it would cost.
I haven't asked that.
There is very, very active competition in oligosynthesis. It's a commodity process. This probably was not very expensive to get done.
Because in the old days it was $1 a base.
Okay. But this is...
They didn't pay $1 a base.
It's gone down.
Yeah, it had to.
And they probably got a quantity discount. So this part was not... actually, this whole paper is actually pretty straightforward.
Yeah.
Yes.
Yeah. Before Rich gets going too far into it, it isn't just synthesizing these DNA fragments for the DNA virus. And there'll be several extra things that they have to do, which I'm sure he's going to describe. But I want to make sure that he describes for us why you can't just put these DNAs together and transfect cells and get virus out like you could with some other viruses. Why is the DNA itself not infectious?
Right. So for most DNA viruses and for a lot of other RNA viruses, you can just make the genome and introduce that into cells, and it will launch the infection. With a DNA virus, for example, like, say, adenovirus, it uses all host enzymes for transcription. So if you stick the DNA into the cells, it goes into the nucleus, uses the host RNA polymerase to transcribe the genome, and that launches the infection. All the other enzymes necessary for replicating the DNA and et cetera get made from that input genome. In the case of pox viruses, you can't do that. The DNA itself, even if you have intact full-length DNA, is not infectious. And that's because pox viruses replicate in the cytoplasm, and because they replicate in the cytoplasm, they encode all of their own messenger RNA synthesis machinery and also all of their own DNA replication machinery. It's the transcription machinery that's the most important. You can't stick a pox virus genome in a cell and have it use the cellular enzymes to transcribe the genome and launch the infection. You have to have some source of enzymes to transcribe the DNA. And so the way this is done...
So in computer terms, there's a bootstrapping problem.
Right. The way this is done, and this has been known for decades, okay? This goes back to old experiments that are called non-genetic reactivation. The way this is done is to infect the cells with one pox virus that then serves as what in the modern age we would call a helper, and then introduce the DNA in on top of that And so the transcription enzymes now have been introduced into the cell. They've been made by the helper virus so that when the new genome comes in on top of that, it can be transcribed by the enzymes that established from the infection that was established by the helper, and then launch the infection and get it going. And it turns out that, okay, so in the old days, it was called non-genetic reactivation. You take one kind of pox virus and infect cells with it, and another inactivated pox virus, and introduce that into the cells and show that it could be reactivated by the infecting helper virus. Ultimately, this evolved into marker rescue, where it was shown, this was used to map mutants and that kind of stuff, where it was shown that you could infect with a virus and transfect in a fragment, and the fragment would recombine with the virus that was infected into the cells, because the recombination in pox viruses is really active. That's all catalyzed by viral enzymes as well. This led to making recombinant pox viruses that can be used as vaccines. So this has been a really robust industry. Now, some years ago, I have the paper here. It's reference 19 in this one. I don't have it. It was like 15 years ago, 2003, I think, as the Evans Lab is interested in recombination, among other things. The Evans Lab published a paper, this is Yao and Evans, where they showed as part of this work that you could actually recreate vaccinia virus by infecting cells with a distantly related helper pox virus that's actually a Leporipox virus, same family as Myxoma, a rabbit pox virus. It's called Shope fibroma virus. You could infect cells with that as a helper, and then introduce on top of that infection a bunch of overlapping fragments from vaccinia. In this case, they were either cloned fragments or generated by PCR. And when you introduce those fragments into the cell, the helper virus would catalyze the recombination amongst all of those fragments and the reactivation of that genome, and you get vaccinia virus out the other end.
Now, it's reasonable to infer from that result that if somebody wanted to reconstruct variola, smallpox virus, they could probably do so with the same technique, right?
Absolutely. It was
so in other words,
a discussion that went around at that point.
Right.
All of the technology was in place at that point.
Right. So in other words, this paper now several years later is causing a similar dust up in, oh my gosh, they showed how you could reactivate smallpox, but in fact, so to speak, the horse was already out of the barn.
Absolutely.
Oof.
And in fact, anybody who worked on pox viruses, even before the Yao and Evans paper, would have said, yeah, you'll be able to do this because of all of the things I've already talked about, all of the marker rescue, all the notion that you can recombine fragments of pox viruses and make recombinant pox viruses in a helper infected cell makes all of this, I don't want to say obvious because that belittles the
work that went into it, right.
the work that went into it. Now, there's one other little thing here, and that's an interesting part of this problem. In that is that pox virus DNA has what historically are called covalently closed ends. It's as if the thing is one big single-stranded DNA circle that is homologous throughout 99.999% of its length, so it makes a double-stranded molecule. When you get out to either end, the DNA continues all the way around in what's called a hairpin. And it's an imperfectly base-paired double-stranded end. And there are two forms of it that in the paper are called fast and slow because they migrate differently on agarose gels. But a close look at those, you realize that if you unfold either one of them, if you unfold both of them, they are complements of each other. Poxvirus DNA replicates as concatamers, that is, several different molecules put together. And in the junctions between the two molecules is the double-stranded form of these hairpin ends. And when the concatemers get resolved, there's a staggered cut that's made on either side of the concatemer junction. And these things, because they're palindromic, snap back on each other to make these hairpins.
You know, Rich, I wish you knew more about this so that you could really inform the audience better.
You know, this is my life.
Yeah, no, I sense your
He's
passion.
had a little experience with this.
I feel your pain.
And those hairpins are absolutely critical for the replication of the genome. Their function in detail is not fully understood. And until modern sequencing techniques, it was difficult to sequence those. There's only been up until now, there's only been a few hairpins actually sequenced, because typically what you had to do, you couldn't really clone these things easily. I think the first ones were done by actually isolating these out of an agarose gel and end labeling them and doing Maxim and Gilbert sequencing. And so most people don't want to go through that trouble. So the horsepox sequence did not include the hairpin ends. All right?
Right.
But the supposition was that there's enough homology amongst these viruses that you could just substitute the vaccinia hairpins and they would work in the replication of horsepox. And so in this particular paper, they had the hairpins synthesized as well using sequence from vaccinia. And so they infected cells with Shope fibromavirus, transfected in the hairpins and all of these other synthetic fragments. And because of the enzymes present in the infection from the Shope infection, there was all of this recombination that went on to zip all these fragments or these overlapping fragments together, and reactivation by transcription of this genome, and they got horsepox out the other end. There are a couple of other intermediate experiments where they show recombination into a vaccinia genome of horsepox fragments, but the real meat of the thing is the reconstruction of horsepox. They had a couple of tricks where in the original, to aid in isolation, in the original synthesis, they included a yellow fluorescent protein gene in the thymidine kinase so that they could pick out fluorescent plaques, because the recombined and reactivated viruses were pretty rare. And then they went back later and recombined that out using the normal horsepox fragments, so they have authentic horsepox.
So, Rich, I had a question. It didn't seem to me from my reading of it that they actually used the thymidine kinase as a positive or negative selection for either of those steps.
They did not.
But they had it in case they needed it, basically.
Thymidine kinase is just a convenient place to put things, because it's not essential. Okay,
I have two other questions at this point. They also put in an AVA1 and a STU1 site in this fragment 2 as potential future cloning sites. And I was just wondering why they put them there, if there's a rationale or not.
If there is, I don't know of it.
Okay. And then my other question is, because they're doing this in the presence of a helper virus, how does this happen so that they're not getting any helper virus DNA in their final virus?
Right. Thank you very much. It's really interesting that, and they discussed this at length in the 2003 paper that describes the technique originally. The supposition is that there's little enough actual nucleic acid homology between Shope and vaccinia or horsepox so that you can't even hybridize fragments in a southern blot.
Oh.
Okay? It's like at least 25% of the nucleotides are different. And it may even be more than that. So the supposition is that the actual nucleic acid homology is low enough so that there is no recombination between the helper and the horsepox virus that you're constructing. The empirical observation is that you sequence this thing and there's no Shope DNA in it that you can detect. It's all horsepox. Okay. And it's probably because of lack of homology. And that is, among other things, why I include that other phylogenetic tree. Mm-hmm. Let's see. This is, pardon me, TWiV fans. All the hosts are all now looking at their document at this phylogenetic tree I put in there. RFV, rabbit fibromavirus. It's in the Monkeypox viruses. Right. You can see is very significantly removed from the orthopox viruses. The other virus that's been used in the same fashion is Followup virus, which is way removed. That's way off in a corner. And in the original paper, Yao and Evans, no, maybe it's in this paper. In one of the two papers, they say that SHOP works a lot better than Followup. It's interesting that the viruses have so little homology that they won't recombine with each other, and yet from a functional point of view are close enough so that the enzymes from one can work on the other. Okay?
Right.
So.
Got horsepox. What did they do with it?
Got horsepox. Now they characterize it. And they do just a few standard things. One is to look at plaque size, and this makes pretty significantly smaller plaques than either cowpox or vaccinia. They do one-step growth curves and show that, interestingly, even though it has small plaques, on most cells it grows about as well as vaccinia and cowpox. The subtlety here is that it also shows very little spread in a plaque assay as opposed to vaccinia or cowpox. And it's also surprising under those circumstances that in a low multiplicity growth curve, it grows as well as it does. But spread's probably important for virulence. And so they tested virulence in a mouse model using different doses of virus to see how pathogenic it was in mice. And it is very significantly reduced in pathogenicity. A dose of 5,000 PFU of vaccinia in a mouse will make them sick enough so that they lose a significant amount of weight. And yet, let me see, 10 to the 7th. So that's 10,000 times as much horsepox, and the mice don't even lose weight and don't have much by way of symptoms. They also show that if they take mice that have been infected with horsepox and allowed to undergo an immune response, that they are resistant to a challenge. So it does a challenge with a lethal challenge of vaccinia. So it does work as a vaccine, as you might expect from all the discussion that's gone on before. So
let me just say something. So this all was done at BSL-2 because this is not a human pathogen, right?
Correct.
So anybody who wants to work on horsepox now can get it, right?
Right.
If these folks are providing it, or you can make it yourself.
I'm sure they'd provide it. Dave Evans is a good guy. Secondly, just a little pedanticism. They say that the indicated doses are known to cause virulence in BALB/c mice. You don't. The dose doesn't cause virulence.
Doesn't cause virulence.
Okay, just a small thing, but it just stuck out. These things stick out at me.
They're known to be virulent.
They're known to be virulent. Yes, they don't cause virulence, right?
Right.
Rich, can you talk about this fact that this horsepox virus synthetically made doesn't produce secondary plaques?
Yeah, so that's the spread issue that I was talking about. If you do a vaccinia plaque assay under liquid, this goes back to, and I have to say, I haven't been this pumped about, I always get pumped about TWiV, but I haven't been quite this pumped since episode 68, Ode to a Plaque.
Right.
Okay?
Right.
And your question goes back to that very episode. Mm-hmm. If you do a plaque assay with vaccinia under a liquid overlay, you find secondary plaques because virus will exit the cells in an enveloped form and then infect other cells and cause secondary infections. In the horsepox infection, you don't see that. So probably most of the spread they're getting is from cell to cell in the monolayer, and they didn't address this in particular, nor did they talk about the genes that might be involved, which are well known. But the implication is that horsepox may not be as efficient as making the form of virus that gains a secondary envelope and is responsible for spread. That's important for the virulence and pathogenicity because any compromise in that spread function, and you make a virus that is less virulent than one that can spread well. So bottom line is, and this is important for the discussion on dual use, which they talk about in the paper that ensues, is that they have used, in effect, existing technology to make a vaccine virus that is of lower pathogenicity than even the standard laboratory prototype virus that is ordinarily used. That's not to belittle the work. It is a technological benchmark, I think, that you can now, even though it would, this is one of these things where you could say, well, it's obvious you could do it. Right. Yeah, it's obvious. So do it. You got to do it.
Is this the first pox virus genome assembled in this way by making DNA?
This is the yes. It's the first assembled in this way with entirely synthetic DNA. I think, among other things, it's a remarkable tribute to the advance in technology that you can make just in DNA synthesis technology, that you can make fragments that are like 10 kb
right.
with accurate sequence. And I was thinking about this. We'll discuss the Dirk issue. I was thinking about the Dirk issue. If you're going to get upset about this paper, you ought to get upset about people making DNA.
Yeah, right. Exactly. Exactly. And so this is dual use research because you could take this approach and use it to make smallpox virus, right?
Right.
And that's why. But in fact,
as we've discussed, that was already feasible.
It was already feasible. So it doesn't really tell you anything that you didn't guess before. They execute it, which is great. But they went through hoops to get this approved at their institution. They talk about that. PLOS also looked at it. They put it on their dual use committee, and there's a Dirk statement from PLOS 1. They have a PLOS dual use research of concern committee, and they were consulted, and they concluded that the study did not provide new information specifically enabling the creation of a smallpox virus, but uses known methods, reagents, and knowledge that have previously been used in the synthesis of other viruses like flu and polio. And so they approved it. And so I think they went through the right procedures, and that's what you should do. And that gets us to this article in NTI where they complain about this paper. They say, as governmental oversight continues to lag behind biotechnology breakthroughs, academic and private stakeholders conducting funding and publishing research must take responsibility for mitigating risk. Well, guess what? They did.
Yeah. Yep.
They did everything they had to. And so this person is calling for more discussion, before these experiments get done. Well, no, we have everything in place to mitigate the risk. And if you don't know that, you shouldn't write this article.
Well, this article is just a fairly shallow blog post, I think, that's intended to highlight one of the top issues for this organization. So NTI is this anti-proliferation group, and they have Elizabeth Cameron, I guess, is in charge of their biological policy programs. And her job is to comment on this sort of thing. So she did. It's not a particularly strong argument, as we've just discussed, but this is the conversation that people are having. Gee, should this be allowed? Well, yeah, we've gotten some useful new information out of that. The thing about the hairpins on the ends, we've got now a less virulent potential vaccine strain. This is, medically a good thing to have. The company's advancing its interests. They're a private company. This is, I think this is totally appropriate.
I don't think it is, because she doesn't realize that there are procedures in place.
The work is appropriate.
I'm sorry. I thought you meant the article was her.
No, the article, calling for more oversight, I don't know what exactly is being asked for here.
Well, she doesn't know either. She thinks there's no oversight.
And that's what I'm saying, is that this is a fairly shallow blog post.
She didn't do the research. Now, what I fear is that someone would pick this up, and this has been in the press, here you go, scientists are making bad viruses again, and that's not the case.
Remember, this is the 100th anniversary of the publication of Frankenstein.
When is it? Today?
Yeah, this is the year.
Frankenstein.
The other complaint you're going to hear about this is that they have exhumed an extinct virus, which First of all, is not entirely true because horsepox does exist with the CDC, and second,
and may exist in the wild. We don't know.
And what it may still exist in the wild, and what they have created is it's a vaccine, right?
Yeah,
it's
great.
It's of no consequence.
That's right.
Yes. Or actually, it's potentially a positive consequence.
Yeah.
It may help us understand the existing vaccines better. It could help us develop other vaccines. And the technology, as the authors point out in the paper, is also going to be useful for engineering pox viruses further in the future because they get used for all kinds of things. Anti-cancer agents, oncolytic agents, potentially vaccines, and et cetera. So the technology itself is important.
I'm sure Tonix Pharmaceuticals is going to do some more preclinical work and see if this is a candidate for a human trial.
Absolutely.
Yeah. It may have far fewer side effects than the existing vaccines, and that would be great. So I don't see how you get scared. We resurrected 1918 influenza virus. People got all upset about that. It hasn't escaped because we know how to contain it. We've gotten great results. Not me, but others have gotten great results working on that virus. And, no problem. It's not easy to do. Not everybody can do this. Iron Man couldn't do this in a cave in the middle of nowhere.
That's correct.
Well, and as I've argued before, I think the risks of this are hugely overblown. If somebody wants to build a weapon and they want to put their energy and their money and their time into building a biological weapon, I don't know. I'd encourage that because there are so many more effective weapons they could be building.
Yeah.
Right? A dozen guys with box cutters knocked down a couple of buildings in New York, killed thousands of people, and completely reconfigured our entire security system worldwide. That's an effective weapon.
Yeah.
This is, it's not going to get anywhere.
Now, let's say in 10 years, polio is eradicated. We have to destroy all our stocks.
Happen.
But it may happen. We have to destroy all our stocks, and that's fine. Will any scientist want to reconstitute polio? No, because we don't need it for anything. We're going to keep the vaccine strains, and we'll have the sequence, of course, but no scientist, who plays by the rules is going to do that. And you can make all the rules you want. You're not going to stop someone who is not a scientist who has evil at their mind from doing this. And so you're just slowing down the scientists by throwing more and more regulations. I think we have all the regulations in place that we need. And I think this is a really cool experiment. It shouldn't generate any Sturm und Drang, right?
Right.
Exactly. Du bist richtig.
What's that?
That means you're right.
Yeah, Rich, anything else you wanted to add? That was very nice.
I think that's about it. Any other questions?
How far away is monkeypox from all this?
Monkeypox is another orthopox virus. It sits kind of between vaccinia and variola. And by the way, it's another, Kathy brought up earlier that how viruses are misnamed based on their isolation. Monkeypox is another example of what is probably a virus of rodents that can infect monkeys, and people out in the wilds who interact with monkeys and or rodents can pick it up. And that's an example that has a certain lethality associated with it. I think it's I don't think it's as much as 10%, but it doesn't transmit very well.
Right.
So that can kill you, but it doesn't transmit very well.
Yeah.
And it's mostly confined to some areas of Africa. You'll recall that there was an outbreak in the U.S. because of imported exotic pets some time ago.
Right.
Yeah.
Luckily, that was a strain of monkeypox that was not as lethal as they can be, and nobody died.
Let's go to picks. How about that?
Picks is good.
Sounds good.
Alan, what do you have?
I have something that actually I think Brianne either put this together or she at least tweeted it. This is a spreadsheet, and it's in read-only mode. So it's a spreadsheet of female virologists. And I'm hoping this is going to be extended with maybe a column saying what they work on. But right now, it's just a list of names and institutions and email addresses. So if you are doing a story, I mean, for reporters, this kind of thing is often very useful. And you're looking for virologists, and you're saying, "Gosh, I'm talking to 3 old white dudes who are all in the state of California. I wonder if I could diversify this story a little bit." And this is a real issue, because if you find yourself talking to a group of people that are all in the same place, you might wonder if you're really getting the full perspective. So this is it's international, and it is being maintained and extended. As I say, Brian would be somebody to get in touch with.
Julie Pfeiffer is the one who
started this. Okay.
Yeah.
And there's
Julie's the one to contact.
And then this,
yes,
there are 2 versions. There's a recent one. I don't know which one this is. I think I picked it on another podcast. And okay. Julie, on Twitter, she periodically puts it out, and there's a recent one like in the last few months. Just see if that's it. I don't remember where I picked it before, but
okay, I bookmarked this, but
was it a long time ago?
When did I bookmark this? 22 days ago.
That's probably the right one.
This is probably it then. All right.
There are 559 names on this. Amazing.
Which also means if you're organizing a conference or finding speakers or looking for people to comment for a story or something, and you say, well, I couldn't find any women in that field, then you're full of it.
Where do these data come from?
Julie put together a list, and then she asked for people on the list to add names to the list, and it just went out from there.
It went viral.
Yes, it went viral.
Yes.
Rich, what do you have?
So I am a member of the PTA for my grandchildren's elementary school, K through 5.
I'm so sorry.
And I joined the PTA among others. Well, I just wanted to support them. But I'm going to participate in their February 8th STEM night.
Oh.
Where they're going to have a bunch of different science and technology and engineering displays out for the kids to mess with. So I decided to do one on microscopy, and I wanted to have a display where it was easy to see what was going to, where you could just look at all kinds of stuff and it was easy to see. And so I'm going to borrow actually a pretty good kid's microscope from one of the parents. That's a regular microscope, but is modified with, this is a double pick here, a digital microscope camera, which is a really cool thing. It's just a little camera
ocular
that fits in the eyepiece and has software to go with it, so that you can project what's in the microscope onto a computer screen.
Cool.
And do all kinds of stuff with it. Because the biggest problem, well, a couple of problems with doing a microscope demonstration. One is that unless you're practiced at it, it's hard to look through a microscope. And the other, especially for little kids. And the other is that unless you've got a fancy teaching microscope, you can't both be looking at the image at the same time and say, look at this, look at that.
Right.
Okay. Well, this way you can put the whole thing on a screen and it solves both those problems.
Yep.
The other pick is a Dino Light digital microscope. And this is a kind of like a little handheld dissecting microscope. So it's relatively low power. You're just looking at stuff that's intact, but it goes up to about 230. I bought this for myself, 230 power, and you can just stick it on anything and have a look. And it actually comes with its own software as well. So you project the image on a computer screen, and you can mess around with it and measure stuff and all that kind of stuff. So I just got it yesterday. I was looking at my skin this morning and grossing myself out, looking at my clothing and anything I can dig up from outside. So we'll dig up a bunch of bugs and maybe see if we can find us some little worms and some pond water and stuff.
Oh, you will. You will.
A look at stuff.
And you should let something rot in the refrigerator.
Yeah.
And do some of that.
I'm sure it happens on a daily basis. He doesn't.
I got some blueberries that are on their way right now.
That's
some cheese.
That's right.
But they have a good demonstration video for this. that I like watching.
Yes,
at
it's
the,
very
at the
good.
bottom of the page. Yeah.
Now, if those blueberries didn't have to come from California and spend so many days, and then by the time you get them, they have 2 days left.
This is
Gee, if they
true.
were hyper local, Dickson,
Hey, you're onto something there, Vincent.
that's cool. I should get one for my lab.
Maybe.
For fun.
Sometimes,
Maybe.
so we lost the ability to take pictures on our tissue culture scope, and so when we have to, I take my iPhone and put it up to one of the eyepieces and try to get the camera to line up. And,
Oh, well, this digital now. If you have a binocular scope,
yeah,
I don't know what will happen with it. How
Well,
the
you get
digital
one.
camera will work.
you get one eye, it's fine. It's just that the resolution isn't so high.
$60, it'll be great.
It's 1.3 megapixels. I don't know if that's good enough for publishing, but it's cheap enough to try.
Too bad.
Dickson, what do you have?
I have a website of a photographer that I recently discovered who spends a lot of his time waist deep in Lake Erie in the winter of all times and photographs the most amazing waves.
Oh, wow.
They are frightening. I mean, I don't want to be anywhere near these events, but I like to see them from afar. And so I was really attracted to his website, and hope you enjoy his photographs.
These are beautiful.
Yeah, this was on my list somewhere. I was
Really?
thinking I'd already
Yep.
picked it, but I guess I haven't. And
Oops.
so it's just still on my list. No, you got it first. It's great.
Dear.
Nope.
Great minds think alike.
There's some of these that look like monsters.
That's
Yeah,
right.
amazing.
They are monsters. If you're in a boat, that's a monster.
The one I'm looking at right now actually has a boat, not a small boat either.
Exactly.
I would not want to be on.
No, me neither.
It's very
Yeah, this is cool stuff.
Kathy, what do you have?
I also picked a photography one. It just came across from the university daily online newsletter kind of thing. It's a photography contest from the Ecology and Evolutionary Biology department here. And so it's a set of photos, and the top two are the winners, but you can click on them and then you get the caption and a little bit of information. So this first one is a venomous frog in Bahia, Brazil. That was the first place winner. And then the second one is also from South America, from the Peruvian Amazon. It's some mushrooms that are just really lovely.
Those are great. Love those pink. They look like cocktail stem glasses.
They do.
They do.
They actually
do.
They do.
Lovely.
Yeah. And then there's some Japanese beetles spooning. And then also I like this one, the I scratch you, you scratch me from Ecuador. The lizards.
That's very cool.
Yes.
I think that's from the Galapagos.
Yeah, it says Galapagos, Ecuador. Yeah.
Those look like the diving iguanas.
Good stuff. Good stuff.
This is cool. It's a Google, doc photo page. You can just download each one to your local thingy if you want.
That's a very rare rhinoceros. That's a black rhinoceros.
Oh, poor cricket.
They're almost extinct.
This is nice.
Very beautiful.
A spider and a cricket.
Gorgeous.
Oh, that's cool.
Yep.
Now this thing here, look at this. Looks like
looking at.
I don't know what this is, but it's yellow glop with little white specks, which look like colonies.
Oh, click on it, and it's just called Party
Crash.
Looks like Dickstelium.
I guess it's a contaminant, right?
It's Dickstelium.
Yeah, it does look like Dicky. Yeah,
It probably is.
And the others are colonies. They're doing some kind of screen because there are lots of colonies, and this contaminated it.
Right.
Yeah.
But, yep. These things happen.
They do.
That's cool. All right, I'm picking my virology lectures for this year, which have just started. It's spring. I teach this course every year to Columbia University undergrads. I record every lecture. It is a principles-based approach based on Principles of Virology, the textbook. And virology.ws/course. You can find PDFs of the slides. You can find study questions that I give my students for each lecture that help them focus on what they should be learning, and the link to the video on YouTube. And eventually there will be 25 lectures, so it'll be complete. And they're 75 minutes long. They're long. I realize that not everyone will watch it all, but they do pretty well on YouTube every year, 20,000 views by the time the year is up. So I think, and a lot of people like them. So here they are again. Tell your friends. And I have a lot of fun. I modify them slightly each year. This year I'm going to I think I'm going to make a new lecture. I'm trying to decide which one to cut. I haven't decided yet, but there will be a new lecture where I'm going to discuss. It's going to be called The Infected Cell, and I'm going to talk about virus effects on the host cell, including, translational shutoff, alteration of membrane, biosynthesis, and metabolism, which we talked about recently.
We did.
That's now at a point where
it should be a Sigma Xi lecture.
A Sigma Xi lecture? What's that?
Sigma Xi is a society, biological society, and they have Sigma Xi lectures that go on all over the country.
I wouldn't want to be a member of any society that took me.
Especially Sigma.
All right, we have 2 listener picks, one from Andrew. This is a very cool pick. Dear TWiV team, given the popularity of TWiV, I suspect you must get endless emails each week from people asking you to discuss their latest paper. Therefore, I won't burden you with another such request. However, a small non-paper nugget that might be of interest is a web server that we made to easily search the data in our latest publication. We generated the complete interferomes, the repertoire of genes up or down regulated in response to interferon for 10 different species, and compared the species. Like other big data studies, this produces a mountain of data that is sometimes difficult to fully grasp. Instead, the web server allows anybody to simply search for any gene or filter based upon certain criteria, then download the results. The web server can be found here, and there's a link, and he had included a screenshot, but it's very nice.
This very cool.
Open data. Orthologous clusters of interferon-stimulated genes. And this is based on a paper by Andrew Shaw as the first author, and it's from Massimo Palmarini's lab at the Center for Virus Research at the University of Glasgow. And so that's nice. Thank you, Andy, very, very much. We'll put a link in, and people can share it.
Cool.
I have a connection with Massimo. He used to be at the University of Georgia, and
that's
my current lab tech here, after I left, worked for him for a while.
That's right.
So,
yeah, when I first met him in Glasgow a number of years ago, he told me he knew you from Georgia. That's right. Mm-hmm. And we have a book suggestion from Neva. In the London Review of Books, a review by Gavin Francis of Pale Rider, The Spanish Flu of 1918 and How It Changed the World, a book by Laura Spinney. Both my maternal grandparents died in the 1918 flu epidemic, orphaning my mother, her sister, and brother, who were parceled out to different aunts to be raised. The enormous epidemic affected populations, nations, families, and individuals on a global scale. This is all Neva writing. I put this book on my reading pile. Neva from Buda. Yeah, it was a big effect. A lot of people died. So you can imagine it broke up families and had serious effects. And just, a virus. All right.
Just a virus.
Just a virus.
Let's give away a book. I'll try and give away a book. Tried once and nobody entered this contest. I must say, I'm a little disappointed that no one tried to do the crossword. I knew it was hard, but you don't have to get everything in a crossword because sometimes the ones you get help, right. Dickson? Yeah. Alan, do you like doing crosswords?
I don't, but my wife is deeply into them. She actually goes to a competition that's organized by Will Shorts, the guy who does the New York Times puzzles.
He's fantastic.
Tell her to try the book one. Not that
every year she goes to the American Crossword Puzzle Tournament, and cool. She hasn't won it, but she does quite well.
Well, the one
where is that? Is that in New York City?
That's they held it in New York for a couple of years. They do it in Connecticut now.
Well, the one I put up,
my wife would be interested in that.
The one I put up was not all that difficult, although you have to know some virology because it has terms in it.
Right.
So Vincent, if somebody had sent in one and it wasn't complete or had a mistake, would they have won the book?
I would give it to them.
Probably would have.
Yeah, sure. If you got it 90% complete, yeah.
Still get an A.
I mean, I want to encourage, the thing is, if you do the puzzle, you're going to think about virology terms, and that's cool. But anyway, now we're going to just I'm going to try another puzzle, which that one was made by Ann Palmenberg at ASV 2012, and it had a lot of meeting-specific, not a lot, but a few clues that were meeting-specific and may have been too hard. So I'll make another one. As I said, I'm making them for my course now, so we'll put one up in the future. But right now, I want to get rid of this book, Neurotropic Viral Infections. This is a brand-new book. I know it's not even been opened. Just I'm looking at the table of contents. And you may say, why didn't you read it, Vincent? Because I have a PDF of it.
Because you were afraid it would get in your head.
I mean, they have chapters on almost every neurotropic virus. These are RNA viruses. There are 2 volumes, one for RNA and one for DNA. If you wanted to buy this on Amazon, it would cost you $239. It's actually more expensive than the DNA virus version of this neurotropic viral infection. Over $200 value.
Well, because they had to reverse transcribe the book.
So it's edited by Carol Schoskis-Reese, who I've known since I was a graduate student. I have a chapter in it, but there are many other chapters as well. Maybe that's why nobody wanted it, because I have a chapter in it. But anyway, all you have to do is send an email to TWiV@microbe.tv with the subject line neurotropic. Just neurotropic. And sometimes people don't put the word in the subject line. I still enter them in the contest. But
you're a nice guy.
No, this time you got to do it. You got to put
do it. Just follow instructions. Neurotropic in the subject line. Exactly. Okay, and we will pick a winner at random. And the whole crossword was done because Islam Hussein said, kind of a shame to just randomly give away a book. Why don't you make people work at it? Well, guess what? They don't want to.
Didn't happen.
Okay. So TWiV@microbe.tv, and you can send us questions and comments. You can send us money if you'd like. You can go to microbe.tv/contribute. Many of you have recently, and we really appreciate it. That's great. Many, but maybe 20 new people over the last few weeks. Greatly appreciated. You helped us to move forward. And subscribe to the show.
Indeed.
If you have a favorite app that you listen to the podcast, just subscribe so you get every episode. Dickson Despommier is at parasiteswithoutborders.com.
Indeed.
Thanks, Dickson. How was the class today? Good?
That was a lot of fun. Yeah, I had a little trouble with the computer, unfortunately. But
I'm so sorry.
We worked it out.
I wasn't there to help you.
Well, someone else had an identical, more up-to-date computer, and we used hers.
Dixon and I went to Brooklyn yesterday.
Yes, we did.
For a couple of hours. We visited Gotham Greens.
We did.
That is the best name for an indoor farm, isn't it?
Yeah.
It's in New York.
Yeah. Yep.
It's on the top of a Whole Foods.
Yeah.
Cool.
There's basically a greenhouse on top of a Whole Foods.
Yep. And
they grow leafy greens in it.
That's right.
And so Dickson interviewed the CEO and CFO and CAO.
The
CAO is the chief agricultural
officer.
That's
right. Cool.
And they gave us some greens. They gave us romaine lettuce and some pesto. It was really fun.
Yep.
Great.
So that'll be up on Urban Agriculture sometime. So Dickson drove me.
He did. Vincent navigated. Along with Siri.
And we drove back on the Brooklyn Bridge. I'd never been over the Brooklyn Bridge. Can you imagine?
Wow.
And we drove past the new Freedom Tower. Is that what it's called?
Yep.
I'd never seen that.
Yep.
Don't really like it.
Yep.
But
what do you mean you've never seen it? You can't not see it.
No, I never drove past it.
You never drove past it. Okay.
Right.
No, I've seen it out the window all the time. Yeah.
Yeah.
But I never drove past
Should go inside. It's an impressive building.
I don't like the top.
Well, write them a letter. Maybe they'll change it for you.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an emeritus professor at the University of Florida in Gainesville, where he used to work on vaccinia virus, although who knows which one or where it came from. He's currently residing in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Filled with fire and brimstone still.
Great stuff.
Alan Dove is at turbidplaque.com. He's on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
And I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the American Society for Microbiology for their support of TWiV and Ronald Jenkins for the introductory music at ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. We have lots of titles.
We do. Horsepox plays well into this.
Yeah.
Yes.
Alan, I think you're the winner again, though. I have to say it, even though I tried hard.
Which one
I cannot do you. A pox on your horse. I love it.
What is the usual thing?
A pox on your house.
Pox on your house.
Oh. Yeah, that's very good.
It's excellent.
Rich, you're the pox guy. What do you like?
That's good.
That's good? Got a lot here.
There are.
And the horse you rode out on.
Right.
Horse poxing around in the lab. That wasn't so bad.
I do like Horsemen of the Apocalypse.
Yeah, that's good too.
That's good. Betting on the Dirk horse is good too, right? They're all good, but we can do a pox on your horse.
The apocalypse, would that be pronounced apocalypse?
I don't know, Kathy. You tell me. Maybe it would be a smooth X, right? It would be a smooth X. That's right.
The other guys don't know what we're talking about. They don't listen to TWiV.
Hey, let's ask Rich.
It's Liza with a Z.
And Alan. There's a word, Axenic. How would you pronounce it?
That's Axenic?
Axenic?
Yeah.
I think that's probably supposed to be a xenic.
You got it.
And Rich, what did you say?
I said Axenic, but I had a clue because of the conversation that just went
right. Well, so on a TWiV we recently, and I say Axenic because I was brought up to say Axenic by all the people who taught me, but it's apparently Axenic is correct.
It means not alien?
That means known life.
No known life. So just if you grow something axenically, you're a bacterium, there's nothing else growing but that one. Yeah.
Like a virus could never grow
axenically.
No.
Right.
Ever.
Axl Rose, I said on TWiP, I think. Axl Rose is not Azel Rose, right?
No.
But the same with, we have these incredible arguments about apoptosis or apoptosis in the book. Yeah. And they think I'm a boor for saying apoptosis. Right? But people can say it both ways. Why not?
And there's also apoptosis. You can get apoplectic about it.
I mean, there's helicopters.
Tell me about it.
It's not helicopters. Helicopters. Right.
Isn't there a poison that you keep mispronouncing also, like amanitin, but it's not amanitin.
Amanitin.
They don't say it that way, though. They say
amanitin.
That's what they say.
So it's amanitin.
Amanita is the fungus.
How do you say the fungus?
Amanita.
I thought it was Amanita. Amanita. Okay.
All right, we got it all wrong.